Hello dear students, welcome to Biochemistry by Revati. I am Revati and in today's class we will deal with the topic Henderson-Hasselbalch Equation, which is a very important and a commonly used formula in biochemistry. It helps us to understand that how the buffer systems maintain the pH levels in the body, means inside the cells and in the blood. Before we understand the equation, let's recall what is actually a buffer. What is a buffer? Buffer is a solution which resists pH changes. It resists pH changes when small amounts of acids or bases are added. Right? And what are the components of buffer? Components of buffer are weak acid and its conjugate base. Now these two work together to keep the pH stable. Now let's look at the Henderson-Hasselbalch equation. What is Henderson-Hasselbalch equation? pH is equal to pKa plus log to the concentration of conjugate base by concentration of weak acid. Let's see what is pH here. It is the hydrogen ion concentration. Hydrogen ion concentration of proton or H plus, right? So it tells us the acidity of the solution. And what is pKa? It is the acid dissociation constant. What is this again? Acid dissociation constant. It tells us how strong or weak the acid is, means whether it is weak or strong acid. Now, what is this? It is the concentration of conjugate base. And finally it is concentration of weak acid. Now, what is the use of this equation? This equation helps us to calculate what? pH of the buffer solution. On ratio of what? Base to acid. Is it clear? Now let's look at the derivation part of the Henderson-Hasselbalch equation, means how it is derived, how the equation is derived. First, we'll start with the dissociation of a weak acid. First, we'll start with the dissociation of a weak acid. What is HA? HA is represented as weak acid. Now, this double arrow represents reversible reaction. Means here HA will dissociate into H plus and A minus. Now, this reversible reaction will shows that the reaction goes both ways, right? So, now here HA is what? It is a weak acid. And what is H plus? It is hydrogen ion concentration or protons. What is A minus? It is conjugate base. Right? Now let's see how the derivation goes forward. First, we have to write the K expression. Right? Ka expression. What is the first step in derivation part? Writing the Ka expression. How to write the K expression? Let's recall. the previous videos, we have already discussed that acid dissociation constant is given by the ratio of products by reactants, right? So what is the product here? HA. Sorry, what is the product here? Concentration of H plus into concentration of A minus. By what is the reactant here? Weak acid, right? Concentration of weak acid. Now this is the Ka expression. Second, we have to rearrange the H plus. Rearranging for concentration of H plus. Let's see. First, we have to write the Ka expression. What is the Ka expression? Ka is equal to concentration of H plus into concentration of A minus by the concentration of Ha. Now, you have to multiply concentration of Ha on both sides. Then what you will get? We get, let's see, Ka into concentration of Ha is equal to concentration of H plus into concentration of A minus by concentration of Ha into concentration of Ha. So here numerator and denominator becoming the same will be strike off. So you will get the equation as Ka into concentration of Ha is equal to concentration of H plus into concentration of A minus. Now again you have to divide by, now divide by concentration of A minus on both sides. So what you will get again? So what is the equation we got before? Ka into concentration of Ha is equal to concentration of H plus into concentration of A minus. Now we have to divide it by A minus, right? So having numerator and denominator same, again concentration of A minus will be striked off. So finally what you will get the expression or equation? Ka is equal to, sorry, Ka into concentration of Ha by concentration of A minus is equal to concentration of H plus. Finally we will get concentration of H plus is equal to Ka into concentration of Ha by concentration of A minus. So this is the second step. What is the third step? Now we have to take negative log on both sides. Let's see what is the equation we got in the previous step. Concentration of H plus is equal to Ka into concentration of Ha by concentration of A minus. Now we have to take minus log on both sides, LHS and RHS. So you will get minus log into concentration of H plus is equal to minus log Ka in, sorry, Ka again minus log into concentration of Ha by concentration of A minus. So in the next step you will get minus log H plus is equal to minus log Ka. In order to keep this minus as plus, we are taking the reciprocal, minus log A minus by Ha, right? So do you remember what is negative logarithm of concentration of hydrogen ions? Do you recall anything? Nothing but pH. So pH is equal to minus log into concentration of H plus. What is pKa? minus log Ka. Now substituting these equations in the previous equation, we'll get final equation as minus log into concentration of H plus, substituting in this equation as pH, right? So pH is equal to minus log Ka as pKa plus log A minus, means concentration of A minus by concentration of weak acid. So this is the Henderson-Hasselbalch equation, is it? So here what is this? pH is equal to pKa plus log to the concentration of conjugate base by concentration of weak acid. Is it clear? Now let's look at what actually this equation means. First, if concentration of A minus is equal to concentration of HA, then log 1 will become 0. So what will happen to the pH? pH will be equal to the pKa, right? So here the buffer solution works best when pH is equal to pKa. Means here when the conjugate base is equal to the concentration of weak acid, right? If in the second case, if the concentration of conjugate base is greater than the concentration of weak acid, then the solution becomes what? It becomes basic. Then what will happen to the pH? pH will be greater than pKa. And the third condition, if you see concentration of weak acid is greater than the concentration of conjugate base, then the solution becomes what? What? It is acidic, right? So here the pH will be with symbol less than pKa, right? So like this Henderson-Hasselbalch equation will tell us how the ratio of base to acid affects the pH, right? Is it clear? That's all for today's video. I hope you have understood the concept clearly. If you have any doubts regarding the concept, please comment on your doubts, right? And if you found this video helpful, please like, share and subscribe to Biochemistry by Revati for more simple and clear explanations. In the next video, we'll discuss the biological importance of Henderson-Hasselbalch equation and the later numerical problems, right? So, until then, keep learning and stay curious. See you in the next class.